Fantastic. Thank you, Natalie, for the invitation, and I'm really happy to be here. So, let's see how we can use this. Yeah, there we go. So just a few words about what we do. It's very similar to the previous speaker. I also work on the genetics of life-threatening infections in children. We're particularly interested in children presenting with the most extreme presentation and trying to understand and proposing a genetic hypothesis. So we really focus on the extremely rare. So I have not worked with acute necrotizing encephalitis patients yet. This is a collaboration that we're embarking on with Natalie and everybody else here. And most of my research has focused on viral encephalitis, particularly herpes encephalitis, which is actually just as rare as acute necrotizing encephalitis. And the tools we have used to try and understand and dissect the genetic basis of this disease, I think is the same approach we want to use for acute necrotizing encephalitis. So I wanted to talk to you about the models that we have used to try and address that in herpes encephalitis, which we will apply to acute necrotizing encephalitis as well. So in the lab, I'm not a medical doctor. I work very closely with my pediatric infectious disease colleagues. I have Dr. Jethro Herberg here. Together we run this pediatric infectious disease clinic at St. Mary's Hospital in London, where it's a monthly clinic where we recruit otherwise healthy children presenting with a life-threatening episode. These children in the NHS in the UK will not be followed up genetically because it's a single episode and does not present as an immune deficiency. And hence we have a research program to be able to recruit these children and carry out genetic testing and figure out what's going on. In the process, we also collect various patient samples. We get serum, blood cells. Sometimes we get skin biopsies because having fibroblasts is very useful for some of our downstream assays. So we can then use them to infect the skin cells, and we can see a very nice readout. From this process, we also create what is the focus of my talk today, which is this induced pluripotent stem cells, which is extremely useful. I'll talk about it in a second. But basically, this will enable us to actually create any type of cell from the patient's blood cells. And that is very useful for diseases that affect tissue types that we can't access. So obviously, the brain isn't we can't biopsy the brain very easily, so this will give us the ability to actually model the brain cells in the lab. Now, these are just the types of patients we have. I have a large interest across various infectious disease phenotypes, but like I said, I'll be drawing from our experience around viral encephalitis. And so very briefly, herpes encephalitis, very rare, 1 in a million cases per year, affecting children upon primary infection with HSV1. And we found in a subset of those children, they carry single gene mutations, kind of like RAMBP2. They don't have RAMBP2 mutations, other types of mutations in an antiviral pathway that affects their ability to produce this very protective type 1 interferon, which is an antiviral protein. So most of what we've done with these patients is taken blood samples and taken skin cells and shown that in their skin cells and blood cells, they have this inability to produce this protective antiviral cytokine. And this is because they have this mutation in the gene, which is critical for producing that protein. So this is just to show you what happens when a patient arrives in our clinic. We get, and very similar to what Sophie and Natalie has been doing, we take a blood sample. From that, we take blood cells. We take the serum and the plasma, which gives us even more information. Where possible, we get a skin biopsy because skin cells, again, is very useful in giving us different types of information. And in some cases, we also take hair follicles. We just pluck the hair, and we grow cells from the follicle of the hair. And these are all, these different cell types can all be used to create this magical induced pluripotent stem cells, which I'll get to in a second. And so what do we do with all this, these cells? We do many things. We we actually infect those cells with the virus. We look under the microscope to see changes in the cell, how much virus is produced. We do plaque assays, which is a specific assay for actually visualizing the virus infection. So the blank spaces you see there are actually the absence of cells, which means that the virus had killed the cells. And then we also look for the immune response. Again, we use microscopy. It's very useful to looking at how the cells respond to the virus. We do cell viability assays. So we look to see how, if the cells die. Susceptible cells to virus infections will die faster. And we also look at the expression of different proteins in the cells. So these are the types of things we do. Now, to the main player of what I'm going to talk today is these induced pluripotent stem cells. So I imagine this is probably not something most people have heard of. It has, so this was discovered by Professor Yamanaka. He's a Japanese researcher who actually won the Nobel Prize for this discovery. And basically what he has been able to show is that you can take as you probably already know, if you take a blood cell you can never put into culture and turn into a brain cell. That just won't happen, or a heart cell won't become a skin cell. So he's come up with a process where you can reprogram these blood cells basically back to the state of the cell when we were embryos. And when we were embryos, when just a couple cells, those cells have the ability to turn into any cell type of the body. And this is what we call pluripotency or pluripotent stem cells. So we can take skin cells, we can take blood cells, and we can now reprogram them using this little process, scientific process, and we can grow them into any type of cell of the body. So basically, we usually take blood cells and we reprogram, and then we can differentiate these cells into the brain cells of our patients. So this is a process that we're going to be using because, as you can imagine, up until now, anything that has to do with the brain, we were never able to show in the patient's neuronal cells, patient's brain cells, that there is a defect in responding to the virus, that there is a problem with responding to the virus. So we've developed this, so we've taken these iPSCs from our patients and we're creating neurons, but we're also creating these 3D structures called cerebral organoids. Some people have called it mini brains. They're not really mini brains. They're small spheroids of what looks like the frontal part of the brain. We can actually create different parts of the brain that way, but this is just a model to try and understand what happens in the brain. So this is obviously very useful because we have an inaccessible tissue type that now we can finally access using the patient's DNA. So it's like their own cells. It is their own cells. And we can use it in various ways. We can also use it to test new genes that we've identified in our patients to see if it's relevant in the context of the brain. And we can also use it as a model to test for different drugs that may not be easily testable. So this work, most of it has been established by my PhD student, Anissa, who has reprogrammed all our patient cells and created neurons, as well as these 3D, organoids, cerebral organoids. And what we do is we create these and we also infect with, once they're established, we infect them with HSV1, which is with the herpes virus, to see their response to infection. So here's just an output of what it looks like. So here you can see that we culture the cells. These are actual cortical neurons from, this is just from a control, just a healthy donor. And we've taken green virus. So this is not infected and this is infected. And you can see in infected neurons, you'll see green. And then we basically look for, we stain the cells for different markers that tells us whether it is a neuron or not. And by doing so, we can actually also see the structure of the cells. And what we notice is that actually using certain markers of neuronal cell markers, we can see that infection actually changes the shape of the neuronal cells, suggesting that there is a cell death. there are different ways we can do this. And we can also do other assays to try to quantitate the amount of virus that comes out of this once you've infected the cells. We can do some fancy things like light sheet microscopy, which is now we have these 3D organoids. We can actually image in 3D where the virus and how the virus has innervated into the brain. And we can actually look at also the presence of, say, your marker, your gene of interest. So in case of acute necrotizing encephalitis, we can stain the organoids for a RAMBP2 and see where it is versus where the virus is to see how it's interacting. And so this is just, in this one, we only see part of the brain just because the laser on the one side was inactivated, but the green indicates where the virus is. And because we infect these organoids it just sits in a soup of virus. The virus goes in from the outside. So you'll see green on the surface. Pink is MAP2, which is just neuronal marker staining. So it's telling you that all the pink cells are actually all your neurons. So you can imagine we do this in a time course. You can see the spread of infection and how that changes. So this is also very useful in us trying to model the viral dynamics and the viral dynamics of the infection during the process. Great. So basically, very briefly, it takes about 40 days it takes about a month to create the iPS cells. And once we have the iPS cells, well, actually, even before that, let's say, no, it's about a month. So we take blood, we have blood from the patient. It takes about a month to create these induced pluripotent stem cells. We then take those, and then it takes about 40 days before we can create this cerebral organoids. And initially, the iPS cells, they look like this. They're just little flat cells, doesn't look anything quite interesting. And with time, as we add different growth factors, they form these little balls of cells. And they are microscopic. I mean, they're a millimeter in diameter. So you can see them, we put them in a 96-well plate, you just see little dots. And we can culture these till 60 days, in which case they do grow to about 3 millimeters. So it's actually visible by eye. So here is an example of where we infected these organoids. And again, we see green virus and infects very nicely. And we also see staining for neurons. So it tells us that these are truly these organoids. And we also able to quantitate the virus. So I'm just going to run through an example of a patient that we've seen in our clinic. This is a baby who had herpes encephalitis and very unfortunately did not survive. And then the mother who also had herpes encephalitis. And so this is just sort of the process of how we go through in the clinic. We took blood where we extract DNA and do whole exome sequencing, exactly the same as the previous talk. We look for different genes. From the plasma part of the blood, we actually test for autoantibodies. In a viral infection, there are many reasons why you can get sick. One of the hypotheses is that you can have an antibody that you make yourself that attacks your own cells. So we look for autoantibodies to exclude that this is the reason why they were sick. We then take the cells from the blood and we test their ability to produce this protective antiviral protein. Where possible, we also get a skin biopsy where we then grow out cells. That takes about a month to 2 months to get the actual cells, and then we can test the patient's skin cells. And this is important because sometimes the virus that you're using doesn't infect your blood cells very well. So skin cells usually are infected by most viruses. So this is a very useful model. We also then create iPS cells. We do it from the blood cells. So we take a little bit of blood and then we reprogram them, create the iPS cells, and we can do cortical neurons and cerebral organoids. So very quickly, I'll run through this. We tested whether the patients, this is the child and the mother, this is the mother's data, and we basically excluded that they had any autoantibodies. So that's the first thing we were able to exclude. Next thing, we checked their blood cells' ability to produce these protective antiviral interferons. and we add various factors to induce that pathway, as well as infect the cells with herpes virus. And we see that actually, in this case, completely normal response compared to a control. So we can exclude that they have any sort of primary defect in producing these antiviral interferons. We then had fibroblasts from the patient, so we did a similar assay where we infected the cells as well as used various stimulants of the antiviral response. And again, in this patient, we found that it was completely normal. And let me tell you, these assays in a subset of patients, we've shown that this pathway is actually important for protection. So a lot of our patients actually don't respond in this pathway. In this particular case, we went down our sort of list of to-do list, and actually there was no problem. Everything looked normal. So normally we would have stopped here and said, okay, and we've also did the genetic analysis and didn't find anything in the list of genes that we already know have an impact. So this would, in terms of research, we would put this on hold, tell the patient we haven't found anything, but research is ongoing and we'll see what happens. Now, because now we have the ability to do this IPS work, we were able to do these next step, which has revealed something quite different. So here we were able to take the patient-derived iPS cells. These are neurons that we created from the patient cells. And here is a healthy donor, and here's a patient. So again, we're looking at green virus. Now, if you just look at the green, it looks like actually the patient doesn't seem to get infected so much because there's way less green. But in fact, if you look at this staining, beta-3 tubulin stains the whole neuron. So it looks, you can actually see the shape of the whole neuron. It also indicates sort of the healthy state of the cells. Healthy cells look like this. They're very sort of stringly and spindly. And you can see in the patient, they're just dots, which tells us that these are all dead cells. So these patients' cells were extremely susceptible to the virus infection and had extremely high cell mortality. So the patient's neuronal cells showed a complete deficiency in able to control HSV1 infection. Here then we've created the cerebral organoids from the patient. Just on the left panel, we show that these are truly neuronal containing organoids from the patient. And here we have the control donor, and then we have the patient. Here we see the green virus. Now this time we see, maybe you can appreciate, there's a much more intense green staining in the patient's organoids. And we can quantitate this, and we were able to show again that there is much higher viral load in the patient compared to the control. The other qualitative difference we can assess from these organoids, which is actually extremely striking and really a visual confirmation, is that these organoids I showed you are these balls of cells. When you infect with HSV1, you do get disintegration of the organoid in terms of the structure. You can sort of see it. In the control, we don't see it so much because they can control the virus. But in the patient cells, the organoid disintegrates. And in fact, in some cases, if we leave it, this is 24 hours, if we leave it another 24 hours, the organoid disappears. I mean, to the point where I had to ask my students, did you actually drop the plate? Did you lose the organoid? So the virus actually destroys the entire organoid. So it's a very clear indication. So the bottom line is, the addition of this type of patient model really helps us in identifying sort of areas where you may have just a gene or a pathway that's only expressed in the brain cells. And because there was no other way previously to assess that, it would usually be missed, or we would have to stop our investigations there. So this really helps us in trying to understand where to look next. And so now we've refocused our efforts on looking at genes in these patients that are expressed primarily in these cells, and we can also do more further work to try and identify which genes and pathways are important in this particular patient. So you can imagine the idea is to use this exact same approach with acute necrotizing encephalitis patients to be able to model the disease. And very, very briefly, can also use these organoids and cortical neurons to test different drugs in the context of in the neuronal setting. So in this case, we looked at the effect of adding dexamethasone, which is an immunosuppressant. This is a steroid, which is also obviously used in treatment of acute necrotizing encephalitis, to show whether the addition of this, in addition to the acyclovir, which is the antiviral drug usually used, can help outcomes. And in this particular case, we actually show that it does help outcome compared to acyclovir alone. And actually, it's not so clear here because the image is not so nice, but basically the cells are much happier. You have high cell viability, and when you add dexamethasone. So it's just another way we can use these organoids to address a different aspect of disease. So that's it. I just want to highlight all the people involved in enabling this type of research. We have all the clinicians here who obviously are at the front line identifying the patients, obviously the patients and the families who contribute to and are part of this study. We have research assistants and a research nurse who obviously enable the process of sampling. We have computational biologists who help in the analysis of all the genetic data. We have molecular biologists in the lab carrying out all the molecular studies. We have a clinical study manager making sure that these studies are done ethically and run properly. And we also rely on a clinical geneticist to report back all these findings and to guide the patients forward. So that's it. I just want to thank again all the people who contributed to this, and mainly the patients and families who contributed. And I'm very open to any questions you may have. Thank you.
Thanks very much. Very interesting. Any questions? That was really fascinating. Excuse my voice. Done too much talking. Will this be able to be used to research things like the timing of treatment and for other styles of therapy like hypothermic things like that? They're a bit more not pharmacological. Will that be something that can be used?
Sorry, if it could be
Like temperature, you mean temperature?
Oh, hypothermic. Okay, yes.
Hypothermic therapies, as well as the timing, delays of optimal treatment times.
Yeah. So I think the thing to keep in mind is that, like all models, this is just a model, and it has its strengths and its weaknesses. The strength of this iPS neuronal cell model is the fact that there is no other model existing right now that can access how the patient's brain cells respond to various stimuli. But these organoids are not a child or a human, so there's so many other components that are missing from this. Primarily, we don't have any blood vessels, there's no vasculature, there is no infiltrating immune cells. So there are all these other components that are missing. So it'll be very hard to model entirely what happens in the course of what a child would go through in life. We can model very precisely, with very precise questions, different things. So like with the drug testing, you can say, well, we know that if we applied a steroid, it would have this impact on the brain cells. Now, steroids also has an impact on the rest of the body that we're not modeling here. So, I mean, for hypothermia, we could. I mean, actually, there is actually interest in looking at how temperature can change our response to infection. And that's something probably that I don't think many people have done with the organoids yet. So that could be something we do, but that won't necessarily translate directly into practice. It would have to go through many other steps, I think, to be able to say, okay, we saw this effect, in these organoids. Maybe the next thing is to maybe see if in an animal model you recreate that in a whole living organism. And then maybe you can then apply it to a patient setting. But so yeah, it depends on the question and how much we know about it and how many other factors would be involved in sort of manifesting that phenotype or that effect. So yeah, I think we just have to be careful about what questions we're asking. Yeah.
I had two questions. The first one was, is this research what our blood work will be used for that we sort of gave yesterday?
I'll probably let Natalie answer. I think Sophie went over a bit of it yesterday, but
so what Vanessa didn't mention is that the whole iPSC work is really costly. It's very expensive, and so at the moment what we did yesterday already is that we're freezing samples that are reserved for this when we get the funding. That's the idea.
yeah, exactly.
Yeah, exactly. Because it is extremely costly and also very labor intensive. So it takes. So you didn't mention. Actually, I might not even know myself, but I think it takes literally someone coming in every single day
yeah.
for 3 months.
Yeah, so I said, you know, what Natalie has taken is she's taken part of the blood cells that you've given and frozen 7 million of them, small vial, specifically for this purpose. We take that when we're ready. We reprogram them. The reprogramming itself takes about 2 weeks. After that, we have to let the cells grow and let these magical iPSCs cells appear. And that is extremely variable per sample. In good cases, in 2 weeks' time, we have nice little colonies that we can just then grow out. In most cases, so if everything goes well, that's 1 month. If it doesn't, it can take up to 3 to 6 months for just getting iPS cells out of one patient. And that whole process means there's someone having to come in every single day to feed them because they're very delicate cells. We can't, if we leave them unfed for one night, we lose them basically. So the whole process takes a while, but once we've got the iPS cells, we can then freeze them and then we can use them for all this downstream experiment. So the first initial part takes time. And yeah, but it's quite labor intensive. And once we start the differentiation, so once we take the iPS cells and say, okay, we're going to make these organoids, it takes 40 to 60 days. Now the first 14 days, again, someone has to be in every single day. And then after the 14 days, it's less of an issue. So you can come in, it's less hands-on. But it is a very labor intensive thing. We're actually also looking to we're working with bioengineers to make this a bit more streamlined so it's not so labor intensive, and there is a huge push from the biomedical field to use this technology in a more usable way because it is quite clunky right now.
And when with sort of both types of research, when would we expect to see some sort of outcome sort of either way from the work?
I was answering this with breakfast to some people. So we literally started experiments yesterday, last night, and we'll do again today for all the people giving blood today. And we'll start getting results in the next weeks, for some things. So next week's coming months, certainly by the end of the year, we'll have something finalized. But the analysis, for example, the whole exome sequencing analysis, several weeks or possibly months, because it's quite complicated. We hope to wrap things up this year, but certainly the whole iPSC part will be the next step. And as Vanessa mentioned, it will open up completely new opportunities because it helps us to access these inaccessible cells, which we have been, obviously we can't access the brain, and we know that we have to look at the brain. So we'll get some answers this year, and hopefully we get the grants to do this part of the work, which will provide more answers after that.
You mentioned the dependency of funding. It would be good to understand more about that. You know, what role, if any, we could play. We're not sure if this is all done sort of behind closed doors, and it's not something that we should worry about, or if we should also invest time and take some responsibility for that side of things.
You could answer too, but I mean, I think, funding, there are many sources of funding for this type of research. And I guess we go to grant, government agencies or rare disease charities also provide this funding. And I guess patient-led charities are a huge part of the work that we do because this is a lot of that, money actually comes to us and we can enable this work. I don't know how that's managed. I don't know if there's someone else who can speak to that a bit better, but I think, yeah, or I don't know, Natalie, if you want to add in anything to that.
Yeah, I think it sounds like it's a big hurdle, but it's part of our job.
Yeah,
We
exactly.
have lots all the time. It's in our DNA, we just do it all the time.
Yeah.
We get rejected, we go again, we
Yeah,
just keep doing
yeah.
it. And this sort of work couldn't be financed by a patient.
Yeah.
Also, because I think it's 15,000
per line. I mean, and that's just to create the cell line. That's not even to cover the experiment. Yeah.
But there's loads of agencies, both national and international, that provide funding for that, especially for rare diseases. And so we've been applying for that. So it's pretty hopeful.
Yes, exactly. Yeah. Thank you. Yeah.
There we go. How important antibodies? So
Yeah.
you look at interferon.
Yeah.
Have you looked at other interleukins like IL-17 or do you do that at all?
So not for herpes encephalitis because there was no indication that those pathways would be involved. So even with herpes encephalitis, there's no case study yet, but all the literature coming out of the autoantibodies to type 1 interferons has come out of many of the vector-borne encephalitis models have it. So we're looking at it just in case. So no, we haven't. I mean, I think my collaborator has done a sort of systematic search for all autoantibodies from plasma from our HSC patients and didn't find anything really. So I don't think that's, yeah, we haven't really, we don't do it because we don't feel that there's been enough evidence to look for it. Yeah. Oh, thank you.
And my DNAs were there. You said the immune genes. Yeah, you'd be able to actually see what's going on with neurons and in case it's just the neurons.
Yeah.
And so, do you have a hint what pathway that's specific to neurons that cause these disabilities? And as well as, do you think that's herpes specific or actually like other things?
Yeah, so basically, so you're asking if it's herpes specific or there's something else you said, sorry.
Yeah, like what
Pathways. So in our model, we are really testing herpes specific pathways, and we've taken all our patients who have the single gene deficiencies, and we're applying that. We're using their cells to actually understand how, if you don't have this gene, what happens? How is it different? But our patients all have mutations in the same pathway. So we believe it is, we don't see from an observational point of view, these patients don't develop any other viral infections. So it's very specific to HSV1. So we're trying to understand sort of that specificity. We actually think it has to do with some cell intrinsic immunity. So the neurons itself has ability to actually fend off infection. It's not infiltrating immune cells. That's why it's so important we're using the organoids that don't have the immune cells. And the other thing we've also noticed is that in neuronal cells, actually autophagy is really important. So autophagy is a process of self-eating. This is something that happens in all our cells. And upon virus infection, this process is upregulated. And in control cells, so healthy people, this happens. Neuronal cells can't self-destruct after infection to protect against further infection, which happens to our peripherals, our blood cells. So neuronal cells use this other mechanism of self-feeding. So they take up the virus or they take up the inflammatory components and try to digest them down. And that's how they get rid of inflammation. So we see that our patients with some of the genetic deficiencies have problems in autophagy. So they can't activate autophagy upon infection, which means they're then left with all this inflammation they can't deal with, and that then leads to necrosis. So we think that that pathway is probably, I don't think that's specific to HSV1. It's probably something that happens if they were exposed to some other trigger that would cause that, another virus infection, it probably would have an impact as well. So we think autophagy is very important in neurons in terms of clearing out excess inflammation. So actually, we've done some studies using rapamycin, which is a autophagy, it's an mTOR inhibitor, but it's also an inducer of autophagy, and it completely clears infection. And also the cells completely happy, which we haven't been able to test for inflammatory markers yet, but we believe that there is a reversal of that. So we're trying to understand how that works. And that's completely different because what we're doing from these patients, we have fibroblasts, we have their blood cells, we have their cortical neurons, and we have their organoids. We only see this effect in the organoids. That drug doesn't do anything. There's no effect on all the other cells, but in the organoid, we see an impact. So again, I think it just tells you that there is, it's worth testing it in the model that it's in. The other thing I wanted to bring in is we're also going to be doing co-cultures with microglial cells. So microglials are very important. They're the macrophages of our brain. It's not in our organoids because that's not part of, it doesn't come from there. It comes from our immune cells. So we're now also doing a culture with the microglials with these cells to see how that then changes the response. So I guess the long answer was just, yes, some of the things we're looking at is very HSV1 specific, but the outputs and sort of the messages we're getting probably has much broader implications to other viral diseases. I mean, viral disease and CNS specific disease, brain specific disease.